So the flow of genetic information within a eukaryotic cell involves protein synthesis, which involves the forming of proteins based on information found in DNA and carried out by RNA. So here we have a eukaryotic cell. This is the nucleus, and this is the DNA, and this is where transcription takes place inside the nucleus, and this is RNA. And then the same RNA strand moves on to the cytoplasm for translation, and the end result is a protein, which you can see here. And before we begin, it's important to understand the differences between DNA and RNA. So DNA contains the sugar deoxyribose, which you can see by the marshmallows here. And RNA contains the sugar ribose, which is the brown, licorice. And then RNA is also single-stranded, while DNA is double-stranded, and RNA is much shorter than DNA. And RNA contains the nitrogenous base uracil in replacement for thymine. So translation begins, transcription is the process by which genetic instructions are transcribed into RNA. And it begins when RNA polymerase binds to a promoter, which is a specific sequence of DNA. So if these two strands, these are the two DNA strands, if they were together, then the RNA polymerase would bind, and then...
And after the RNA polymerase breaks the hydrogen bonds between the nucleotides, RNA comes in and forms hydrogen bonds with the DNA.
However, by nucleotides individually.
The nucleotides individually. The phosphates and the riboses are connected by covalent bonds. Now thymine is replaced with uracil because RNA doesn't contain thymine. It only contains uracil. And what happens is RNA copies information from DNA, and this strand comes off. Hydrogen bonds are broken. This strand of DNA comes back, the nucleotides...
This happens when RNA polymerase reaches a termination signal.
And then after the termination signal, the DNA nucleotides then reattach via hydrogen bonds. And the phosphates and the...
Okay, so the RNA then moves on to these two, the DNA come together.
The RNA goes to the cytosol to begin translation.
Goes to cytoplasm to begin the translation. So it's important to keep in mind the transition from the nucleus, which you can see here, to the cytoplasm, which is represented by the orange board. So translation is the process of making a protein, and proteins are made up of polypeptides, which are made up of chains of amino acids connected by peptide bonds. So translation begins when tRNA and mRNA join together, and enzymes attach an amino acid to one side of the tRNA, which you can see here by the pressure, and then the other side contains the anticodon that's complementary to the codon on the mRNA. So the start codon is AUG, and that codes for the amino acid methionine, and its anticodon is UAC, which you can represent it by these bases here. So they will pair up like this. And then the ribosome, our model basically shows progression. So the ribosome would first start here, and then the next tRNA containing the amino acid glycine and the anticodon would pair up with the next codon. And then once this happens, the ribosome detaches methionine from the tRNA, and then the peptide bonds forms between the two amino acids, which you can see by the green candy. And then the first tRNA leaves the ribosome, and the ribosome then moves on to the next second codon. And this process continues until it reaches the stop codon.
A stop codon, which is either UAG, UAA, or UGA.
Yeah, and we chose to represent it by UAA. And then once it reaches the stop codon, the elements of translation come apart. The newly formed polypeptide falls off.
And they start to fold.
The ribosome moves away from the mRNA, and then the last tRNA the last tRNA leaves the ribosome. And then eventually the amino acids will form to form the end result, which is a protein here.